Hello. In this video we're going to introduce some data that will be used for a coding demo. So the coding demo we're going to look at is R programming, what I want to do 5 minutes of coding, at most 10 lines, and see what we can do. R is a really good language for dealing with tabular data. So think of things that you would open in Excel, but we can handle lots of it. And do nice statistics and make nice looking graphs and visualizations. And I just want to see what can we do with 5 minutes of coding and 10 lines. But first I want to introduce the data to you and where it comes from. Okay, so this is a scientific paper where they're explaining where what the experiments are that we're going to look at the data. Okay, so this is in the area of trying to understand heart development. And this is a graphic of different samples that were taken in this study from mice. So you have a whole bunch of mice that are allowed to grow. So they start off as embryos. At day 7, they're going to take some of the embryos and measure the gene expression of all the different genes. That's what this icon here is supposed to mean. So at day 7 they take the whole embryo. At day 8 they take the heart from some number of the mice and measure the gene expression. At day 9, also day 12, 14, 18, 3 days after birth and adult. And they also took some mouse stem cells and measured the gene expression of those. And what we want to understand is how do the genes change throughout the development of the heart. Okay, so we want to look at all these genes and we want to look at how do they change. So are there some genes that are important at this kind of phase here where the heart is developing? This picture here is a heart tube. So first you have a heart tube, and then it sort of starts to become more like a mature heart. So what are the different genes that are important at the different stages? All right, so we're going to take a look at how this data looks. So I'll load this data in R and take a look at it. So we have 45,101 rows here. Each row is a particular gene. The first row here is the COPG1 gene. Okay, and then each column, we have some columns here just saying what the genes are, and then we have 3 columns here for stem cells. So what that means is there's 3 different times that 3 different replicates, samples that they took of the stem cells and measured the gene expression of all the different genes. This 9.422 is a number where if it's higher, then you have more of that gene active, and if it's lower, you have less of that gene active. So if we look at the stem cell versus the embryo, it kind of looks like they're at about the same level. Then if we go up to day 8, this one is maybe slightly higher. So we've got the day 8, 3 different samples there. And then when we get to day 9 and later, there's 3 samples for the left ventricle and 3 samples for the right ventricle. So those particular parts of the heart, going to day 12 and adult. Those are the ones that we're going to look at. Okay, so the question that we want to look at is, in this data, which of the genes show the biggest changes at the key time points? There's 45,101 genes, and we want to see what can we do to figure that out with not much R code and not much time. So that's the challenge, and we'll do that in the next video.